Yo no sabía mucho inglés cuando iba yo sabía decir, ok, y tú. Y mi profesor era chino, que hablaba. No, no es que estos chinos hablan el inglés bastante más rápido. No sé, profesor, usted que conoce personas de otros países, ¿verdad que sí?
I think so.
Yo siento que lo hablo muy rápido. Y a veces
But the thing is, I'm used to like a lot of accents because growing up and in different labs that I've been in, I've heard English in different ways, so I can understand it pretty easily.
todo el mundo.
All right, so let's get started. So just before we get into the actual seminars for today, we have two visitors. We can welcome them to the class. Hello, thank you for joining us. So today, what I want to briefly mention is, so as you know, I rejected the rough drafts because they don't follow what I had asked for. So what I want to emphasize is that your manuscripts, regardless if it's a rough draft or the final draft, it has to be an actual complete composition. What that means is that you need to tell the full story. For the rough drafts, I understood that was not going to be complete. So I don't necessarily expect all of the ideas to be fully fleshed out, but I do expect that there be a story that I can actually give you some guidance as to this story being developed properly, or you might need a little bit more context, whatever. But in the current form, there was no way that I could do that for either draft. So I gave it back to you, obviously with the intent that you add in that context. I understand that it's not going to be a complete draft, so my goal is not that by Friday you have the complete paper. What I want to see is that there's an introduction and I have an idea of how you're trying to connect these ideas. That's what I want to see for Friday. Another thing to emphasize, and I sent you an email of examples of the previous papers. Now, obviously, those are polished. What I mean by they have gone through several rounds of editing. So I don't expect that. But I do want you to get an idea as to how different themes connect and how you might try to create a coherent voice. That's what I want you to learn from those examples. And then remember that I recorded the discussions about annotated bibliography because there were some things that I had emphasized that I want to see in the manuscripts. So look back at that lecture. I didn't post it online. I sent it to you by email. Look back at that lecture because I gave some key ideas that I want to see in the papers and how I can, the different people who might be able to contribute to that. All right, so with that being said, today we're going to have a seminar that's going to focus on the application of viruses. And so we're going to have Carmen and Yolmari both going to be external, right? So Carmen is off in France and Yolmari is in different part of Puerto Rico. And so we're going to have this hybrid presentation where they're going to talk about a more traditional perspective of how metal complexes are being utilized with respect to a potential antiviral application. And then I'm going to transition into how we can possibly weaponize viruses with metals to try to have therapeutic applications. So different perspectives in that regard. So we're going to start off with Carmen and Yolmari, so you guys can take over. Carmen, are you talking?
I'm not. I think Yomari was supposed to start, but she's muted.
Okay.
Oh, sorry. Hi, everyone. Today, Carmen and I will be giving a seminar on the antiviral metal complexes. And for this purpose, we selected the following article: Gold-based metal drugs as inhibitors of coronavirus proteins, the inhibition of SARS-CoV-2 main protease by aronophine and its analogs. You can pass the slide. So in the context of COVID-19, as we are well aware of, that has become a global health problem impacting both our society and our economy. And research efforts have been focused mainly on the development of new treatments, hopefully to be able to control the spread and reduce the severity of the virus. Drug exploration has been mainly focused on synthesizing and repurposing organic compounds. However, inorganic approaches have not been as extensively studied or explored as the organic compounds, but due to the success of gold-based metal drugs that they exhibit in treatment of other diseases, then they may be able to become potential candidates for antiviral applications, and it is definitely an alternative worth exploring. Change the slide. So main protease is a homodimeric protein with each monomer consisting of 3 domains, and its active site is going to be localized between domain 1 and domain 2, particularly cysteine 145 and 156 are 2 of the 3 residues that are going to be exposed to the surface of main protease. It functions as an enzyme that basically is going to cleave those viral polyproteins into functional proteins that are required for the virus to replicate. And so therefore, main protease is a drug target of COVID-19 because it plays a role on the virus's replication.
So one of the things that, particularly in this paper, we are talking about is the use of gold-based drugs. And this is because gold-based drugs have been known to interact with the sulfur-containing targets that you see oftentimes in these adducts, in these amino acids. And one of the focuses in this paper is on Aranaphen, due to the fact that it was originally used as a rheumatoid arthritis drug. So this has already been FDA approved. It's already under investigation as a cancer therapy. It already has some cytotoxicity studies, and it's really just repurposing this drug to be used as an antibacterial, antiparasitic, and now antiviral. And there have been some studies on the use of Aranaphen as an antiviral through computational modeling, but not a lot has been studied. And other methods of inhibiting viral proteases with gold have been studied, but most of the metals that have been studied are not gold. And gold wasn't really a metal that was studied for inhibition and antiviral inhibition until COVID-19. And part of that is due to some of these studies that were performed through computational modeling. And some of these previews have shown that gold targets the cysteine residues that Yomari was talking about, and that we've seen the crystal structure of an adduct between the M-Pro and Aranaphen previously showing that it does bind to that catalytic cysteine through the gold. The gold does bind to that catalytic cysteine. And this is part of the reason why this is being studied. Additionally, studies have shown that enzymatic fluorescence resonance energy transfer assays have shown that gold does inhibit happen-like proteases, or PL-Pro, of SARS-CoV-1 and SARS-CoV-2. Obviously, SARS-CoV-2, COVID-19 is what we're going to be talking about. And in these studies, it showed that Aranaphen, in the case of SARS-CoV-1, had the lowest inhibition with an IC50 of 25.5 micromolar, but it had a really strong inhibition for SARS-CoV-2 with an IC50 of 1 micromolar. And this is part of the reason why they decided to start focusing on Aranaphen, but they didn't want to just do Aranaphen. They also wanted to look at other small molecule gold complexes that haven't been approved for clinical use yet. These are all complexes that have been extensively studied for cytotoxicity for cancer. A lot of them are under investigation for cancer studies, but we also want to be looking at their antiviral effects and their those kind of abilities that they have. So the researchers chose 3 different halide analogs, which we see here in the gold triethylphosphine, chloride, bromide, and iodide, as well as, and then 2 different gold carbenes in the gold NHC and the gold NHC2 PF6.
So as the overall objective of this study, it aimed to investigate the potential of gold metal-based drugs as inhibitors of coronavirus proteins. specifically the compounds that Carmen just described and the inhibition target that I described at the beginning, which is the main protease, which we're going to be referring to as MPRO. Slide. So one of the methods used was an enzymatic activity assay in which basically a fluorescence labeled substrate is cleaved by the enzyme, and that cleaving releases the fluorescent molecule, and the release of the fluorescence molecule is going to result in an increase of the fluorescence intensity. So in this study, the substrate that the researchers used is specific to the MPRO enzyme. And when the MPRO cleaves it, it's going to result in that increase in fluorescence intensity. And that increase is going to be proportional to the MPRO enzymatic activity. So therefore, we can use it to measure that. So once the gold compounds were added into the reaction mixture, the decrease of the fluorescence intensity can then be used to measure, in turn, their inhibitory activity. Slide. So through the enzymatic activity assay, the values of Ki for all of the gold compounds were obtained. The Ki, which is the inhibition constant, basically measures the binding affinity of an inhibitor to its enzyme. And the relation is that the lower the Ki, the more tightly it binds to the enzyme, and therefore more efficiently it inhibits its activity. And so it turns out to be that the most potent inhibitor of MPRO was the oranophine halide analog containing bromine, because the rest of the compounds did show inhibitory potency, but not as much as the halide analog containing bromine. And with an exception of these, the gold NHC, PF6, turned out to be a very weak inhibitor, so it's not even included here. And it inhibited less than 10% the loss of activity, even when they tried it with high concentrations as high as 60 micromolar. It still did not, it was still a weak inhibitor of the MPRO activity. Next slide. So these graphs represent the percentages of MPRO activity and its relation to the concentration of the gold compounds. So the inhibitory activity was manifested to be carried out in a concentration-dependent manner, because with an increase in inhibitory concentration, there are more chances for it to be able to bind to the enzyme's active sites. And again, we can see that all exhibit inhibition activity, but the halide analog of oranophine containing bromine is the most potent one. Next.
So one of the other things that the researchers did in order to really understand how these drugs are bonding and if they're bonding is using electrospray ionization mass spectrometry. And we talked about this a little bit in the beginning of the semester, but as a little bit of a recap from then, or reminder, electrospray ionization mass spectrometry is done by applying a high voltage to a liquid to create an aerosol, as we can see in the schematic on the right. And this creates ions from the macromolecule fragments, which can then be processed through mass spectrometry. And this will show you what fragmentation, how the complexes are fragmenting and what fragments are available. So the results that I'm about to show you in a little bit are showing that the MPRO does bind the metallic fragments from the gold drugs, and we're going to see two signals, the mono and the bis adducts. And the bis adducts are actually the more common adducts, whereas the mono are more infrequent. Again, and additionally, most of these fragments correspond to the gold triethylphosphine ion, and this is mostly due to the fact that this is part of the oranophane molecule, and that's most of the analogs that we have. So to start out this work, the researchers performed a blank with the MPRO by itself without any sort of coordination to see where it fragments and how that looks. And then they use that to compare all of the other samples. And as we can see, especially in these first couple, you can still have that free MPRO peak showing that not all of the MPRO is bound and is coordinated. So we're actually going to have the BIS adduct in the center, this higher peak, and the mono here. And we can see that this is the gold triethylphosphine ion, and this is one of the most common ions that we see. Again, for in part C, we can see the protein binding through the sulfur and the gold of the NHC carbene ring. And from the lower intensity, we can see there's a poor reactivity, but it does still bind, and there is still something happening, and there is still binding of that MPRO. In the next set, we have the ranofin analogs with the halides, particularly in these two, bromide and iodide. And again, there is the BIS and the mono adduct, and in the case of the iodide, there's only the BIS adduct. Finally, in the chloride analog, we can see again the BIS and the mono. And I also took the gold NHC2 PF6 mass spec to show you what it would look like if something wasn't bonding, if nothing was fragmenting. And here they saw that there was no metallation of the MPRO even after 24 hours of incubation, while all of the other analogs and all the other gold-based drugs have metallation after 4 hours. So this is just not really binding, and it's thought to be due to too strong of a bond between those carbons and the gold, and it's not letting the gold go and bind with the MPRO itself. Following this, or alongside of this, the researchers attempted to grow crystals of the analogs with the MPRO. And anyone who's really done protein crystallography will be aware of the fact that this is not an easy thing to accomplish. And they were able to grow crystals with the bromide aranofin analog, as well as the gold NHCCl, and those structures were solved. They found that these adducts both resemble the apoprotein, and they both show low gold occupancy where the original organic moieties are no longer visible in the crystal structure. So starting with the gold NHCCl system and the gold NHCCl crystal, we can see that the gold is binding to that catalytic cysteine 145 with an occupancy of 0.15. The gold sulfur bond is 2.4 angstroms, which is in line with what you would expect for a coordination to occur. Additionally, they found that there's a labile water molecule that's coordinating with the gold ion itself, which was an interesting discovery, but they don't really talk about how that affects anything or if that affects anything. It seems like that might just be part of maybe stability. In terms of the bromide analog, there is 2 binding sites with the cysteine 145, that's that catalytic cysteine, and the cysteine 156. Starting with cysteine 156, this binding site shows really good occupancy of 0.4, and it binds to the sulfur of the cysteine, but it also, as you can see, has a second bond at the terminal nitrogen of the lysine 104. So this is a bidentate gold binding of the MPRO itself. And then when you get to the catalytic cysteine 145, there is a significantly lower occupancy at 0.15, but this is almost identical to the binding to the occupancy previously seen with the gold CHN Cl molecule, and this shows a weaker electron density and a monodentate coordination. So both of these structures support the conclusion that there is inhibition of the MPRO due to the binding of the catalytic cysteine 145. And this is really important for what we know and what we can use for inhibition in the future and for these structures themselves.
So to conclude, the gold-based drugs, auranofin and its analogs, were able to inhibit the pro-enzymatic activity as shown from the low inhibition constants that they presented. Also, the lack of a gold NHC2PF6 adduct formation accounted for its weak inhibition, and it is perhaps due to the strong gold carbon bonding, as Carmen was explaining. In addition to that, the electrospray ionization mass spectrometry data presented the formation of stable metallodrugs protein atoms. It was also found that gold fragments coordinated to accessible protein residues in 1 or 2 locations. And finally, based on the crystallographic data, it explained the gold coordination to cysteine 145 and cysteine 156, which I mentioned at the beginning. And therefore, it supports the conclusion that the inhibition of MPRO is due to this binding.
So one of the biggest study limitations that we see in this study is the fact that there are no in vivo studies performed showing the gold complex's actual inability to inhibit SARS-CoV-2. So what that is, I think, one of the biggest limitations. So we're saying that, yes, it will likely inhibit it, but because none of these studies were performed, we're not sure. And so future work in this would be to do those in vivo studies and really see how that works. And I think also it would be really interesting to see a greater catalog of gold complexes with MPRO inhibition, because as I mentioned towards the beginning, there have been a number of modeling studies showing a number of gold complexes that can inhibit PL-PRO, which is the happen-like protease, and that seems to behave similarly to the MPRO. So it'd be interesting to see how some of those other complexes that do well in that protease perform with MPRO inhibition. Finally, it would be really interesting to see continued studies of the use of auranofin and other gold complexes as antivirals in general. As I mentioned towards the beginning, this work with gold complexes and gold-based drugs as antivirals is fairly new. Most of the articles that you're seeing, most of the research that you're seeing has come in the last 4 to 5 years, and a lot of it is due to the COVID-19 pandemic. So it'd be interesting to see how these complexes work with other viruses and as antivirals in other ways. So thank you very much for listening. And if you have any questions, we'd be happy to answer them.
All right, thank you. So we're going to start with questions. All right, so Javier, any questions?
Yes. Great presentation you do. Can you go to the crystallography part? So, yeah. When I was reading the paper, and you mentioned it there that the organic molecules are not seen in the crystal structure. Do you think this can affect probably like there's some degradation of the molecule of the metal complex, and that's when you only see the gold complex? Because compared to the MS data, the whole molecule is seen. However, in the crystallographic part, we do not observe that organic moiety.
So this is something that I also noticed and was surprised that they didn't really talk about. I'm actually not sure. I would expect, I guess, degradation of the organic moiety, or perhaps it's not crystalline. They did report a parameter that talks about the disorder of some surrounding molecules, but I didn't include that data in here because other papers that I read showed that that specific parameter isn't very reproducible. And so it's possible that the organic molecules are just too disordered to see, maybe. I'm not really sure, and I do think more work on that would be interesting.
Thank you.
So before we move on to much, just to touch upon what Javier was asking. So I think what's really important, Carmen and Yolmari, about the crystallography is the crystallization conditions, because they tend to be quite distinct for proteins, whereas when you're assessing for mass spectrometry, where you definitely have to remove quite a bit of salt to minimize the amount of ions in your background, you're going to have a different ballgame of like what's in the actual solution mixture. So in crystallography, you're using things like polymers to try to get the protein to actually crystallize, come out of solution. So there are a lot of things there that can result in potential ligand dissociation, whereas in the electrospray, you're basically removing many different things that could potentially compete with the complex itself. So that those are different factors as far as what constitutes the conditions for crystallography and what constitutes for mass spectrometry. All right, Alex, any question?
She was thinking of other like complexes that have been seen to have similar activity. I would like to know a little more.
Yeah, so you had mentioned about other complexes with this particular protein
Yeah.
that have an interaction, inhibitory interaction. Can you give a little bit more insight about that?
So most of the other complexes that I saw weren't actually with M-Pro. They were with PL-Pro, which is a similar protein, but not the same. I don't know exactly what their structures look like off the top of my head, but I have seen, and I think in the beginning in some of those modeling studies, I have the links to, I would have to double check, some modeling screenings that were done for gold complex inhibition. Some of them were aranofin like.
Oscar?
Yes. Para mí el título quedó bastante inconcluso en términos de análisis inhibitorio, porque lo que yo leí algunos artículos en comparación para este mismo artículo y vi casi diferentes tipos de análisis. ¿Ustedes faltaría parte del programa? Porque para mí faltaría un effective maximum concentration for the drug as effect, the inhibition parameter, and two, the value for between the inhibitory concentration and maximum effect concentration. And this work and the supplementary data using this data.
Did you hear that, Carmen? Yolmari?
I heard parts of it, but not quite the whole thing. Sorry.
Okay, so what Oscar was asking or giving insight about, he felt that there was a bit of inconclusiveness as to the inhibition of these complexes. So he was wondering if you could give a little bit more discussion about what additional characterizations can be done to assess better this inhibition. And so what was the examples?
The half maximum effective concentration, AC53, and the radio for both is that typical for all these compounds for both. They're using for the study for specifically for these alcoholism que he visto en otro artículo con el mismo, pero tenían esos otros elementos que este no tenía.
Okay, so he was asking about EC50, which is similar in line with like what you were talking about inhibition, and then the other one was the ratio.
The ratio and between the two es una comparación en 60 unidades para ver la efectividad.
So you're talking about stoichiometry there, or what are you talking about there? What are you talking about there? Like, I'm a little bit uncertain. Digo en español.
Ellos además de calcular cuánto era la máxima concentración permitida para el obligatorio, el parámetro que es 50, ellos estudian el radio entre lo que acabo de mencionar contra el máximo inhibitory concentration. Y hacían un radio para ver cuánto efectiva era la droga en comparación de la concentración máxima permitida con la inhibición general. Y ahí te explicaban una data donde te explicaban IC50, AC50 y J-radio, que así te explicaba mejor la efectividad de las drogas compuestas a base de oro y también de orofin.
Oh, so tú estás hablando de the metal itself, right? And el tamaño del metal, is that what you're referring to? And how it inhibits the cysteine side, is that what you're referring to?
I don't know, because it's only placed cuál es la concentración máxima, porque esto aquí es la máxima inhibición concentration.
But you're talking about radius, right?
The radius
Of the metal.
entre la razón entre ambos parámetros. The radio. Yeah, sorry.
Okay. Between EC50 and IC50. Okay. All right. So obviously they didn't do that, but so Carmen, he was just saying that in other studies, he has seen an assessment of a comparison of the ratio between EC50 and IC50. So he was just wondering if you were to, further this study, what would like to do?
Porque antes de hacer lo que ellos dicen que quieren hacer en un plan futuro, para mí sería antes hacer lo que ellos mencionan.
It's okay if you don't know. It's just it's a question.
Yeah, I'm not really sure. If people, I mean, I don't know a lot about inhibition studies, mostly what I've read through this article and we were looking up through the article. If it's common for people to do EC studies and then do the ratio of the IC to EC, and that will give you a better idea of the inhibition, I think that's a great idea. I do know that some of the studies were talking about the use of gold specifically because of hard soft acid-base metal theory. And I'm not sure if that applies to what your question is. I just kind of heard the size of the metal mentioned, and that's part of that, I believe.
That came from me because I missed
Oh,
something.
okay.
But that's actually a good direction, Carmen, because I was wondering about that. Just like types of soft metals, and you're trying to fit a certain pocket, right? So you have a certain space of how to accommodate the gold and the ligand. So I was just wondering about that. You don't necessarily have to answer that. I was just wondering how much space is there to accommodate the metal and even some of these ligands. That's going to be an important factor. But yeah, certainly changing out the gold for like, I don't know, like silver or something like that, and what impact that could potentially have in terms of an inhibitory effect. But I do have a question. Oh, go ahead, Carmen. I didn't mean to cut you off.
I do know that some of the articles that I was looking at for using metals to inhibit viruses were talking about other metals like, I believe, titanium and manganese, other kinds of metals like this, and they have more use or more, not use, but research done on them for the same use.
Brenda, any question? Brenda has no question. Javier has a question.
Well, it's not a question, it's more of like.
Comment.
Well, when they listen data, they observe that the only one that's just not bind is the one with the biggest counter ion. So probably the selection of counter ions is very important in order to optimize this kind of possible inhibition for label complexes, right?
So that's what Javier was just emphasizing, that it looks like the counter ion size might be contributing also to what may potentially inhibit or not inhibit. So like, which was the counter ion?
That was
F6,
I believe. Let me pull up the structure here.
Yeah, so that phosphine, yeah, so PF6. Yeah, so that potentially is an important feature as well. Yeah. The size of the compound plus the actual counter ion might have an influence on this.
And the binding between the ligand and the metal is also, as we can see, pretty important if they're thinking that the gold is too strongly bound to those carbons, which is might be part of the reason why it wasn't inhibiting very well. Because in all the others, that halide is the leaving group, which allows the gold to bind.
So remind me, Carmen, I don't remember. So in the electrospray part, did they see that compound always fully intact? The one with the two NCH ligands?
So the two NCH ligands, they don't really show, there it is.
Oh yeah.
Any sort of fragmentation.
Yeah, that's fine. Yeah, I was mentioning the did they do the assessment of the small compounds, not with the protein? To see
They didn't say. Yeah.
Okay. Yeah, I was just wondering about that because sometimes the mass spec can give you some idea about the relative lability of those ligands, or even if they dissociate. Jalen,
It might have been in the full spectra, but it seems like they probably cut it to show just what they want to show.
Right. Yeah.
Yeah.
Jalen?
Yo no tengo preguntas sobre el artículo, una versión, pero sí se me hace curioso que esa misma falta que nosotros vemos cuando no leemos, que hicieron el estudio, ellos lo saben. Cuando hacen estas comunicaciones, yo pienso que no las completan, porque como son temas que son por mejor. No sé si así que se lo dicen. Yo no sé si es la necesidad de comunicarlo rápido, que eso hacen las cosas como fragmentadas. Y es bien bonito hacer la historia completa, hacer algo bien hecho, publicarlo algo rápido.
Sí. So,
La necesidad de conocer más.
right.
Exacto.
So yeah, Carmen, on your mind, Jayleen was just commenting on sort of the limited scope of the paper that, the main focus was just showing how this compound or the GOAT specifically is inhibiting this protein without necessarily going into the in vivo part, maybe as an intent to just get that information out. And they're conscious of the limits of what they're actually showing. The one thing I was going to say to you, because I know a little bit about some of the work that was done as far as assessing GOAT. So you said you would have liked to see some in vivo studies. So I do believe, if I'm not mistaken, this is building off work where they showed infection of the virus in host cells. I believe it was Vero cells that are pretty good, standard cells that they're used for infections. I think Aranafin had been shown to inhibit the infection of those hosts with the SARS-CoV-2. So I think this may be building off of that by trying to give a little bit of insight as to how exactly that may be happening. That's my perspective of why they don't show any infectivity studies. All right, so no other questions. Another round of applause for Yulmari and Carmen. I'm going to take over the sharing.
Yeah.
Actually, before I do this, let me get myself ready. Just want to get that started. I'll get back to it so you can actually see them. All right, so what I would like to present is looking at how you can potentially use viruses for an application that is an anti-cancer application. And in this context, what we're going to look at is those viruses that fall in the category of oncolytic viruses and how you potentially can synergize them with metals that have a known therapeutic property. So here we're focusing on the use of vanadium, which, as we discussed in the class, we have mostly focused on the potential vanadium for an anti-type 2 diabetes application due to its insulin-enhancing function. But there have been studies assessing vanadium for an anti-cancer potential and how that might be useful in this perspective of utilizing viruses. So this article, multimodal potentiation of oncolytic viral therapy by vanadium compounds. So this was done by Debbie Kranz and collaborators. All right, so I wanted to show this video here to just give us a little bit of perspective. I remember Alex a week ago asked me a little bit about this. So I wanted to share this with you.
The immune system is a vital tool in the body's fight against cancer. Despite its efficiency, tumors can evade the immune system by inhibiting the body's ability to recognize and eliminate them. These unrecognized tumors can then grow uncontrollably, requiring innovative treatment approaches to both stop the growth and make them recognizable again to the immune system. Oncolytic viruses are a novel and elegant approach designed to harness the body's own immune system to attack cancer cells by detecting previously unrecognized tumor cells and exploiting their inherent cancerous properties. This new class of cancer therapeutics uses native or genetically modified viruses designed to selectively kill tumor cells. Oncolytic viruses, theoretically, have the versatile potential to attack multiple types of cancers. These viruses are designed to selectively replicate in tumors, causing cancer cell death with the goal of leaving healthy cells unharmed. Tumor cell death, also known as lysis, could result in replicated viruses that can travel to and destroy neighboring tumor cells. This process is intended to elicit a specific anti-tumor immune response by stimulating the innate and adaptive immune systems. Oncolytic virus-mediated tumor cell lysis is thought to release tumor-derived antigens as well as danger signals that trigger an innate immune response and the dual signal activation of tumor-directed T cells. The tumor-derived antigens are then thought to recruit and activate antigen-presenting cells, which prime T cells to differentiate into both effector and memory T cells. This adaptive immunity could result in an army of effector and memory T cells that can return to infiltrate and favorably alter the tumor microenvironment by making the tumor cells more susceptible to immune attack and by recruiting additional immune effector cells. Increased numbers of T cell infiltrates activated against tumor-derived antigens could help educate the body's immune system to recognize and attack previously unrecognized tumors. This newly inflamed tumor microenvironment could make the tumor more susceptible to the body's robust anti-tumor immune response.
Okay. So I wanted to show you that perspective so that we can understand what are oncolytic viruses, because they can be natural, but they can also be engineered to try to make them more selective towards cancer cells. All right, so these are viruses that are preferentially infect and kill cancer cells, and they do so by, as we saw, inducing the lysis of these cells, so basically bursting them open. And as a consequence of that, they result in the release of these infection virions that can destroy the remaining tumor. The viruses also operate by stimulating anti-tumor immune responses by basically labeling the cell surface so that now that surface is tagged and it can be recognized by the immune system. So now you're making the immune system now be recognized, whereas a lot of cancer cells have the ability to evade the immune system. So several oncolytic viruses, or OVs as the abbreviation, demonstrate efficacy in preclinical models of cancer and in clinical trials. The US FDA and European Medicines Agency, EMA, approved TBEC and Lytgic for the treatment of melanoma. So it's been observed that this particular oncolytic virus synergizes well in preclinical models with immune checkpoint inhibitors. So essentially, these inhibitors help to strengthen the immune response. So core infections of tumors can be a major limitation and a factor in the resistance to oncolytic viral therapy. Obviously, not everyone's going to respond to this type of treatment, which is true for many different types of treatment when it comes to the complexities of cancer. Pharmacological compounds can help block the cellular innate antiviral immune response mediated by type 1 interferon, IFN. So here, vanadium compounds can potentially be useful. So we've talked about vanadase, and we looked at different vanadium compounds from the perspective of them serving as PTP or protein tyrosine phosphatase inhibitors. And that's as a consequence of their ability to mimic phosphate. So we looked at them in the perspective of inhibiting PTP1B as a way of enhancing insulin. So several vanadium compounds exhibit an anti-cancer effect. And so this is something that mechanistically is still being elucidated. But it's also been shown that vanadium compounds have the capacity to impact the immune system or trigger an inflammatory response, which then has an immune consequence. So this mechanism by which the vanadium-based compounds can modulate anti-cancer immunity is also something that's being elucidated. So given their potential immunostimulatory effects, several select vanadium, and I say vanadium-5, but I should have been more careful. There was even vanadium-3 and vanadium-4 assessed in this paper. Compounds may serve as a complementary pharmacologic agents to oncolytic viral-mediated immunotherapy. All right. Obviously, I was not complete here, so you can ignore this part here. Sorry, I meant to go back to the slide. So you can see that I was in between. Anyways, I meant to finish off this slide. Here we're looking at one of the things that they wanted to assess was the ability of vanadate to influence the multiplicity of infection of different oncolytic viruses to identify a virus most enhanced by vanadate. So they wanted to understand what type of viruses it can actually, oncolytic viruses, it can actually modulate its activity, and then to treat different types of cells and basically assess this activity. Then they also wanted to understand, is this something specific to vanadium, or is this a sort of a general metal property? And they wanted to look at different metals As part of that was to also look at whether this is something that phosphate, given that it can mimic phosphate, it can enhance, and then to apply this into an actual animal model and see if this is something that can translate from cellular infection to actual animal infection. So that's the rejection, but obviously I did not finish writing. All right. So the influence of antidote on the growth of a small panel of candidate oncolytic viruses is assessed here. So what we're looking at is different types of oncolytic viruses. So here we have looking at measles, this particular the vasculature stomatitis virus, or VSV Delta 51, Synvis, HSV, and Cinia. So we're looking at these different viruses, and we're looking at what happens to them in the renal cancer cell line 7860, and we're trying to understand the influence that vanity has in terms of the replication of this virus. So essentially, we're looking at conditions where we don't have any vanity, and conditions where we have anywhere between 100 to 150 micromolar. I believe the only one that was 100 micromolar was the measles, with everything else is treated at 150 micromolar. And what we're essentially seeing is the capacity of vanity to potentially increase the amount of the viral load infecting this cancer cell line. And so where they tend to focus a lot on is this virus here, the VSV Delta 51, because of that pronounced ability to enhance the yield at a low MOI. So MOI again is parameter, let's go back to the slide here, is the ratio of virus to host cell. So it can influence how much it replicates even at a very low level. So that's what they decided to really focus on this virus. So throughout the paper, what we're going to look at is the response of this virus as seen in different cell lines. And so we are going to see a few assessments being done with particular 786 renal carcinoma cells. And looking at here, how there's a dose response in terms of enhancing the replication of this virus in a cancer cell line that is generally resistant towards the replication of this virus. So we're looking at this in terms of the parameter titer, and we're looking at the dose response of vanadate. And we see that there is a response that tends to basically maximize in this concentration range, which is typically the concentration that you see used throughout the paper. And then when you go too high of a concentration, then you see that it drops off. So that's basically that the maximal level of vanadate that will give you an optimal replication of this particular viral load in these cells. And so then what we're assessing here then is looking at how vanadate can increase the viral expression in cells that are of a cancer nature. So we're looking at these two particular cell types, the CT26WT, which is a colon cancer cell line, and DBT, which is a brain cancer cell line. And we're seeing the basically expression of this virus as measured by the use of the green finger protein. So you can see that, green fluorescence protein. You can see that fluorescence is an indicator of the expression of that particular virus. And as you can see, in the presence of vanadate, okay, again, in this concentration range of 100 to 150 micromolar, you see a predominance of that viral replication in those particular cancer cells where you don't see it happening in these other tissues that are not of a cancerous nature. Right, so that gives you that perspective of the selectivity of this virus for cancerous tissue, but also how the vanadate helps to enhance that replication. All right. So one of the things that they really want to assess is this something that's particular to vanadium? And so what we're looking at here is a series of studies that give us some insight as to whether other metals can actually influence the replication of this particular virus in the 7860 cells. And so we're looking at molybdenum, tungsten, manganese, chromium, aluminum, and two different types of aluminum. And what we generally see is that there's really not any dose response, for the exception of that chromium, for whatever reason, at that 10 micromolar concentration, there's an uptick in a significant uptick in the amount of viral expression, but that's then quickly, just doubling that concentration, we see it drop back down to the standard level. So generally speaking, this suggests that these metals are not influencing that the expression of that virus. And so, particularly for the molybdenum and tungsten, those two metals tend to mimic some of the properties of vanadium in a different context. So that's why those particular metals were utilized. And you see that they also are not influencing anything here. So as I mentioned to you, given that vanadate is in other contexts, it serves as a phosphate mimic, the question was, well then, is the phosphate able to mimic this property of vanadium? And so as we can see here, and using different sources of phosphate, essentially phosphate with different coordination states at different concentration, we don't see any increase in the replication of that virus. All right, so this does appear to be something that is specific to vanadium, and here we're looking at different perspectives, where if you look at the figure to the left, we're looking at different sources of vanadium, where we have orthovanadate, metavanadate, we have other types of vanadium-5, and even that modulato complexes that was tested for an anti-cancer, so the anti-diabetic purpose in reached clinical trials. And so you can see all these sources of vanadium do exhibit a dose response enhancement of the viral replication. And then when you look at vanadium salts, vanadium-3 and vanadium-4, we also see a similar effect, maybe not as pronounced, particularly for the vanadium-3, where you definitely see that there is this property that seems to be quite specific to vanadium. All right. So another thing that was assessed was the pH dependence of this property. And so we're looking at vanadate. We've already discussed how complex that aqueous speciation can be, right? So when you're looking at vanadium-5 in water, you're going to get a lot of these molten vanadium centers. They tend to be vanadium oxide. They're quite complex, but even still, you basically have a fairly consistent profile of how the vanadium can impact the replication of this virus, and it doesn't seem to be very pH specific. So it's something that's very clearly not pH independent. All right, so here we're looking at then another way of how you can assess whether this is really due to the vanadium itself. So what they decided to do here was to use ascorbic acid and a ligand called tyron. Tyron is a catechol-based ligand. And so essentially we want to see by the use of chelation, are we able to inhibit the capacity of the vanadium to exhibit this property of enhancing the replication of the virus? And so as we would imagine, you can see that in the treatment of these chelators, you do suppress the amount of any enhancement of this replication of the virus. So very clearly, the accessibility of the vanadium is crucial to this property. All right. So the next thing was to understand that this capacity to induce cell death. So what we're looking at here is the viability of these cells and how they're influenced by the presence of the vanadate, okay, in one regard, and also in the presence of the virus plus the vanadate. So what we're looking at here is we're looking at the vanadate co-infection with a low MOI of the VSV delta 51. And so we're tracking, I forgot to put the cell, I believe it's the very same cell line, if I'm not mistaken. And we're tracking the percent viability based on dose response. And if you notice that we're focusing on the vanadate, so that's the one in blue, that the dose response is pretty minor, and you have to go into really high concentrations of vanadate to be able to see any pronounced inhibition or of the actual proliferation of these cells. And so you up to about 1 millimolar, this is where 1 millimolar, you're basically reaching an IC50 level. So that's a pretty poor indicator of activity of just the vanadium itself. But when you have the co-treatment or the co-infection, you want to call it from that perspective, the virus with the vanadate, now you can see that that's getting shifted significantly. Where now you can have the IC50, if we use that line there as a gauge, where now we're dropping this significantly from 1 millimolar to about 31 micromolar. So you can see that enhancement in terms of the potential effect on the cell line. Then looking at the panel B, what we're looking at here is a flow cytometry experiment where we're trying to assess what is the mechanism of potential cell death, right? So the figure on the left was telling us that the virus with the vanity has this capacity to inhibit proliferation, right? But the figure on the right gives us a perspective of cells dying and also what is the mechanism of death. And in this context, then, what is it telling us, based on this profile here, described in terms of these panels, that you have an induction of apoptosis. So the combination of both is giving a very pronounced apoptotic effect. All right, so the next question was to see if the cytotoxicity is something that is independent of the actual virus itself. And so we're looking at the percent viability under these different conditions. So we're looking at conditions in which we have no infection, and then we have the presence and absence of vanity. And so we can see there that 30 micromolar vanity drops the viability, but in a fairly minor way, just like we saw in the previous slide. But then we're looking at in the presence of the virus itself, and you can see how significantly that viability decreases. Now we're basically almost at zero there. But then we wanted to see what happens if you have the virus, but in two different treatments. So one of these treatments is by basically inactivating the virus. And so surprisingly, you also see this effect there. And then the other treatment is by doing a mutation, which is basically affecting the replication of the virus itself. And that does seem to have an effect that's going to be an increase in the viability. So there's a less of an effect with the vanadium and the virus itself. So for whatever reason, the UV inactivation didn't have that capability. Right. So they wanted to then get some insight as to what's happening mechanistically. And it turns out that the vanity seems to be increasing cytokine-induced death. So cytokine is a part of the immune system. So it's a response to, a potential anti-inflammatory effect that the vanity is triggering. So what we're looking at here are the different cytokines and what effect it's having in terms of the anti-proliferation effect of the vanity. So again, we're assessing the viability in the absence of the virus. So we just have the 47 micromolar vanity. We see that there is a drop in terms of the viability. But then when we have these three different cytokines present, we see that becomes enhanced. And so it seems that this is something that is a contribution of, a correlation between what the vanity is doing and as a consequence of these cytokines in the presence of the virus. Because you see that there's no effect when there is no in the presence of the virus with the vanity, because you can see that there's no effect when you just have the virus in the absence of the vanity. All right. So the next question, or the next thing they want to assess, is something that we talked about: the capacity of vanadium to trigger the formation of reactive oxygen species, and how that might influence the activity with regards to its cytotoxicity. So we did mention that one of the limitations that vanadium can have for an anti-diabetes application is this parameter, that it can induce ROS. And so that's what's being assessed here. And what we're looking at is the different conditions that can influence the viability of the cells in the presence of vanity. So we're looking at in the presence and absence of the actual, I think this is the treatment here, that we have in the in the presence of those different cytokines. And then at the very last parameter they're looking at is in the presence of treatment with the N-acetylcysteine, or NAC, which is something that's used as an antioxidant. And so the idea was that this is going to give us a perspective of whether there's some contribution of ROS formation. So if you use antioxidant, you're going to minimize the amount of ROS that's being produced. And so you should help these cells to be more viable. And that's what was observed here, that there is an increase in the viability of these cells in the presence of the antioxidant. And so we also want to see how that may or may not influence this ability of the vanity to influence the replication of the virus. And you can see in the presence and absence of the NAC, it has no impact on the capacity of vanadium to trigger that replication of the virus. So NAC is only affecting the capacity of the vanity to trigger ROS, but it does not affect its ability to enhance that viral replication. as you can see here. It's basically statistically insignificant there. All right, so they then wanted to assess these properties, but now in an in vivo model. So what they wanted to look at was, can the vanadate enhance the oncolytic activity of this virus in vivo? And they were focusing on the BALB-C mice. So they were looking at 6-week-old female mice. And so these were given subcutaneous tumors by injecting syngeneic or immune competent cancer cells. So syngeneic means that they were given tumors or cancer cells that were of a rodent or origin, which in this case was of a mouse origin. And then after a few days of tumor growth, the mice were then treated by an intratumoral injection of the vanadate. And after a few hours, the SV delta 51 expressing luciferase was injected intratumorally. So they purposely use this form of the virus so that they can be able to track it. So luciferase is something you can actually image. And so what we're looking at here is this mouse model, and we're looking at these different types of tumors that are being injected. So we have the CT26WT, which is, I believe that was the colon one, the 4T1, which I believe is a brain one, and the DBT. I might be getting them a little bit backwards here, which is the renal one. So they're looking at these cancer cells that were implanted into the mice, and then they're looking at what happens when you have the vanadate and also the virus. And so what we can see here at the bottom is the different responses and what influence you have here above just the background. So we have in blue, which is a little bit hard to see here. The blue is the indicator of just the virus alone, okay, and how much is actually being expressed, and from this perspective by gauging the luciferase, and we have this color pattern here to give us the gradient of how much expression there is. And so then we're looking at with vanadate, sorry, without vanadate, and then in the presence of vanadate at a dosage of 40 milligrams per kilogram of animal. And so we can see what influences it's having, and seems to be pronounced with the CT26WT, not so much with the 4T1, and then some amount with the DBT. So that's something that seems to be cancer specific in this regard. All right, so another perspective was then to look at how this is affecting the percent survival. I'm not sure that's very serious. So we're looking at percent survival with the different types of tumors that have been implanted into these mice. And then we're looking at conditions of PBS, that's the blue. Then we have the vanadate treatment, 40 mg per kilogram. Then we have the dash blue. So the dark red line is the vanadate. The dash blue is the virus. And then the dash red is the virus with the vanadate at 40 mg per kilogram. And so what we can see here is how the percentage survival is altered, and how the vanadate plus the virus is helping to improve survival. So in the case of the CT26WT tumor, we see that we have an enhancement of survival. So now we have like some amount of these animals that do survive. It's not going to be an overwhelming amount, but you can see that the influence of the combination of both. And then you see for the 4T1 and the DBT, a similar pattern, except for the 4T1 at the end, all mice seem to have died. So you can see that, but there is an improvement of how many days to death. And then you have an enhancement of how many animals survived in the DBT case. But here, it seems to be something that is not so specific to the presence of both the vanadate. Well, excuse me, I'm going to say again. You see a pronouncement right here. So that was the most effective one. And does that correlate to what we had observed over here in terms of the effect of DBT and CT26WT? And to some extent it does. But you see that in this expression of the virus, both DBT and CT26WT had more of that replication in the presence of the vanadate itself. And you kind of see that reflected in the case of the DBT and the CT26WT in terms of you do increase not just the how many days after treatment or after the treatment of these mice with the tumors, but how many actually survive. All right. So another really interesting thing is that the idea is that you want to trigger an immune response. So oftentimes when you're trying to trigger immune responses so that you can have a long-term effect. So they wanted to assess that. if you have already treated a mice in this context where these potentially you're triggering because of the use of the virus with the vanadium, you're probably developing antibodies against this type of cancer. Does this hold true when you try to reinfect or you try to place these cancer cell lines back into the mice? Will they grow again? So what they did here was to treat these CT26WT, these were the colon tumor cured mice, and they wanted to see if you try to re-implant that in them, do these cells grow? And what was observed was that they seem to have become cancer resistant. So you see that you have basically 100% mice survival because they were not able to grow the tumors, and as a consequence, all the mice were able to survive. So there seems to be some amount of effectiveness in terms of an immune longer-term response. And so another thing that they wanted to assess was this ability of injecting the vanadate intratumorally, and how that might still have a capacity to trigger an immune effect that might be able to reach to a distal site. So here, what they did was you have the tumor in both the left and the right flank, but only the right flank was injected intratumorally with the virus and with the vanadate. And so what was observed was that, in fact, if you look at the expression of the virus as imaged by the luciferase, you see, like, what's happening there in the presence of the vanadate. But really importantly, we can look at it from this perspective. By using these calipers, you can measure the size of the tumor, and what you can see is that in the presence of the vanadate plus the virus, you can see that the right flank, for all these different mice, you see that the size of the right flank basically shrinks. And then you see that the left flank for several of these are also getting impacted. Whereas you don't see this as pronounced in the condition of the vanadate or even just with the virus alone. So it's a combination of the virus with the vanadate has that very pronounced effect where the site of injection, but also even in the distal site, it's probably triggering some antibodies that can have this response in this site. All right, so they wanted to understand, well, how exactly is the vanadate doing this? And so what they were focusing on was the known property of vanadate to trigger a pro-inflammatory response. As it turns out, vanadate inhibited a type 1 interferon response in 786-0 cells, but potentiated a pro-inflammatory response via type 2 interferon. The data below are presented as a heat map showing the expression levels of the differentially expressed IFN stimulated genes. Expression of genes was normalized to values obtained for mock-treated infected control. All right, so how are we actually looking at this? So what we're looking at is the expression of these particular genes, or at least the levels of these genes. And we're looking at the condition of just mock, which is just treatment typically with PBS, vanadate, the virus itself, and then we're looking at the vanadate plus the virus. And what we're assessing is the amount of those genes that are associated with type 1 interferon and the amount that are associated with type 2 interferon. And so what we're seeing here is that we have this amount of the levels of the type 1 interferon, but then when you have the vanadate plus the virus, you see that the general level is lower. So that's why it's registering as red, because you're getting an inhibition of that. Whereas for the type 2, this is where the level is at in the presence of the virus, but then when you have the virus with the vanadate, you see that these levels are increasing. So that's triggering in this heat map as green because you're actually going above the level from where the standard is. So that indicates that this is the response from which is an immune response as a consequence of that vanadate and how it's influencing the effect of the viral attack. All right, so as an overall conclusion, the authors demonstrated the potential vanadium compounds in enhancing oncolytic anti-cancer therapy. The vanadium-induced OV-enhanced growth can be related to vanadate binding to phosphatase, although that part of the paper isn't well fleshed out. So that's where we know that property exists, but it's not as clear in terms of the overall impact that the vanadium is actually having. The vanadate-induced ROS in promoting a death may be initiated by type 1 IFN. ROS inhibition decreases vanadate cytotoxicity, but does not affect the growth of the oncolytic virus. This work demonstrates the ability of vanadium compounds to convert the antiviral type 1 IFN to a pro-inflammatory type 2 IFN response. This leads to better oncolytic viral spread and a greater anti-tumor immune stimulation. So that's what they were able to assess with this work. All right. Any questions or doubts? Go ahead, hurry up.
Like, it's only the slide, I think, right after the pictures. Shows the performance of the different viruses.
Yeah. Yeah, right.
The HS.
Yeah, so in this case, it actually seems to have an opposite effect.
Yeah.
So maybe that for whatever reason, the vanadate at that concentration is maybe having an antiviral effect. That's most likely how we might be able to interpret that.
Yeah, so the other one, the must the opposite of the first frequency. And in terms of the ROS probation, there is a possibility of instead of using because they from what I remember, they didn't use any higher so probably using that. Do you think it would lead to fertilization since we are also talking about it? So
Yeah,
the plants?
that would be interesting. So I would assume, I'm trying to think about the properties of vanadium and how it might be able to lead to that. So the thing about vanadium, so we're talking about vanadium 5 with the vanadate here, but probably realistically in cells, it might be more like vanadium 4. And I'm not sure if that has the capacity to do the lipid peroxidation part. I would have to look at that more closely because that's going to be one of the key components for the true ferroptotic effect, because not just the ROS formation, it's that part.
Yes.
That's where I would have to look at. Yeah.
No hacer la lipid accumulation, right?
Mhm.
No hacer la lipid.
Not that I'm aware of, but that might be something that was not necessarily assessed. So that I would have to look at it in the literature. Other people have looked at that.
Porque estoy pregunta porque me metí con los negras ROS, también se ve una acumulación de lípidos ROS.
Mhm.
Acumulando pues, si quieres,
Yeah, the thing about ROS is like you have to be a little careful because that could even be just apoptotic type, which we saw in this case. So ROS is more of a general parameter, and so you have to see, well, what else is being generated or what else is being triggered besides ROS. Other questions?
La velocidad de señales, si no hay muchas señales de alguna otra información de industria para los.
Yeah, I'll look it up. Not that I'm aware of. All right, so I apologize for my slide objectives being incomplete. That's 11 o'clock at night type of thing. All right, you all experience that too. All right, all right. Take care. Bye bye.